Hello friends, in this lecture we are going to understand the fertilization, pregnancy, placenta and sex determination. And today's lecture we are going to understand about fertilization and little bit about the placenta. So let's get started. The most important thing are what are the questions that are already asked from this very chapter. The first one is the physiological changes during the pregnancy, placenta, the lactation, puberty, pregnancy test and gametogenesis. And these are the other list of a question that is already asked from this very chapter. So pause the video and write it down somewhere. So let's get started. Fertilization. Now what is the fertilization? The word fertilization means that fusion of a haploid chromatin of a male and female gametes. So from the ovum there are haploid number of chromatin, and from male there is a haploid number of chromatin. When they fuse, they make a complete cell having a 46 chromosome. It is a fertilization, right? It is known as zygote. So there are various events are actually taking place one by one in order to do fertilization. So first enumerate the events. First event is that the chemoattraction of a sperm to the ovum, then adherence of sperm to the zona pellucida. Now you know that zona pellucida is the covering of a oocyte, right? Third event is that penetration of a zona pellucida and acrosomal reaction. Then adherence of a sperm head to the cell membrane of the ovum. It is known as olemma. Next one is the breakdown of the area of the fusion, and finally release of a sperm nucleus into the cytoplasm of the ovum. So don't worry if you do not understand this thing, because we are going to cover it one by one very thoroughly. So starting with the first one is the penetration of an egg. So as far as the penetration of the egg or the ovum is concerned, see, once the ovum is ovulated or released or expelled from the ovary, it has a two structure, right? One is the oocyte, then this blue colored covering that is drawn in this diagram, that is zona pellucida, and there are set of a granulosa cell, right? That is the cumulus granulosa cell. It is known as a corona radiata, right? So in order to enter inside this oocyte, the sperm has to penetrate two things. One is the cumulus granulosa cell, it is known as a cumulus ophorus. Second one is to pierce the zona pellucida, right? So sperm has to pass through two membranes before it reaches to the oocyte. One is the outer coat of the cumulus ophorus or the cumulus granulosa cell or the corona radiata. Second one is the inner coat of a zona pellucida. Now penetration of a cumulus ophorus is very simple. If the hyperactive sperm, the hyperactivity of the sperm, it will eventually cross or pierce the layers of a granulosa cell. Second one is that hydrolysis enzyme that is present in the acrosome, it will release and it will dissolve the cumulus ophorus, right? So this is how the sperm get penetrated to the first layer, that is the granulosa cell layer that is present around the zona pellucida of a oocyte, right? Now next layer is the zona pellucida. How the sperm will enter and make a aperture inside zona pellucida. Now this one is very tricky, so keep attention. So this is the beautiful diagram. I taken this diagram from the book named Tortora. So these are the granulosa cell layer. This blue color are the zona pellucida, and this yellow colored region is the space that is situated between zona pellucida and the actual oocyte membrane. That space that is present between the zona pellucida and actual oocyte membrane, it is known as a extracellular space, and there is a particular word to this extracellular space. It is perivitelline space. So this blue color lining is the zona pellucida, and the extracellular space that is present in between the zona pellucida and the oocyte membrane, that is known as olemma. it is known as, this extracellular space is known as perivitelline space. Now first thing first, once the sperm will pierce the granulosa cell layer, then sperm cell has a receptor for ZP3 protein that is present over the zona pellucida, that I told you in the maturation of the sperm that is taking place in the epididymis, right? Sperm get ZP3 receptor. That ZP3 receptor is eventually attached to the ZP3 protein that is situated over the zona pellucida. So sperm, first thing first, it bind with the ZP3 receptor that is present over the zona pellucida, and once the sperm will bind with the ZP3 receptor, acrosomal reaction will start. That acrosomal reaction will eventually release the enzyme known as acrosin. Now once the acrosin release, that acrosin will produce a aperture inside the zona pellucida or penetrating slit. Once the zona pellucida bind with the ZP3 receptor, then it triggers acrosomal reaction. Acrosin will release. That acrosin will eventually make a aperture inside the zona pellucida, and through this aperture the sperm will enter inside this extracellular space. What is the name of this space? It is perivitelline space. Once the sperm will enter inside the perivitelline space, it will bind with the protein that is present over the oocyte membrane or oolemma. The name of that protein is fertilin. Got it? So there are two things that sperm has to bind. One at the zona pellucida by ZP3. Second one is the fertilin protein that is situated over the cell membrane of a oocyte, that is oolemma, right? Now sperm in the oolemma, once it is entered, once it is bind with the fertilin protein, now sperm head will enter with the nucleus along with the sperm head will diffuse inside the cytoplasm of the oocyte. That cytoplasm of the sperm, the cytoplasm of a ovum or the oocyte is known as ooplasm, and sperm tail will disappear, and once the head will eventually enter inside the ooplasm, the head, the nucleus that is present over the top of the head become decondensed. Got it? And once the one sperm entered inside the one oocyte or the ovum, the remaining sperm are not able to enter inside that particular oocyte. That blocking of multiple entry of the sperm inside a single oocyte, it is known as the blocking of polyspermy. Got it? So let's write it down what we understand up to that level. Sperm bind firmly to the zona pellucida by ZP3 receptor that is present over the sperm, and ZP3 protein is actually present over the zona pellucida. Now once the sperm bind with the ZP3, it triggers the release of the acrosin. Now this acrosin will eventually produce a aperture, or it is known as a penetrating slit inside the zona pellucida, and through this slit sperm will enter inside the perivitelline space. Now what is perivitelline space? Again, it is the space that is present between the zona pellucida and oolemma or the oocyte membrane, and sperm bind with the oolemma by protein fertilin. Now once it bind with the protein fertilin, the head of a sperm will eventually enter inside the oocyte. So sperm in the ooplasm, once it is entered inside the ooplasm or the cytoplasm of the oocyte, sperm tail become dislodged and disappear, and nucleus become decondensed. And once this happens, there is a blocking of the polyspermy. Now the meaning of blocking of the polyspermy is to block the multiple entry of a different multiple sperm in single ovum, right? So this is the next understanding how the second sperm entry is blocked. Now it is very interesting where the sperm, or once the one sperm entered inside the ovum, it blocked the entry of the other sperm. So the answer is like that. When sperm membrane fuse with the olemma, see, whenever the sperm membrane fuse with the olemma, over here, it depolarizes the membrane of a oocyte. It depolarizes the olemma. Once it depolarizes the olemma, what it will do? It will release the intracellular calcium in the ovum. It stimulates the release of the intracellular calcium that is stored inside the ovum. It will release, and it promote the exocytosis of the cortical granules that are situated superficially over the olemma. You can see this green color round structure are the actual cortical granules. That cortical granules are eventually will diffuse inside the perivitelline space. You can see in this layer, this granules is eventually poured its substance, and it will block this entire perivitelline space, and this sperm get dislodged. It prevent the entry of this sperm. So these cortical granules are actually already present over the outer border of the oocyte membrane, but once the sperm will attach to the oocyte membrane, it causes the olemma depolarization, right? So once the olemma get depolarized, it will stimulate the release of the calcium from the ovum, and once the calcium is released, then it promotes the exocytosis of the cortical granules that is present superficially with the olemma, and it fills the perivitelline space. So this portion of the perivitelline space is already filled, and this one is still remaining because this sperm this perivitelline space just getting started to fill with this cortical granular substance, right? This is the one thing. Second thing is that whenever this single sperm entered inside this olemma, entered inside this oocyte membrane, the granule that is released, the cortical granules, the content of the cortical granules, it has an enzyme. That enzyme will eventually degrade the receptor that is present over the zona pellucida. Let me repeat one more time. First, once the single sperm entered inside the oocyte membrane, cortical granule will diffuse inside this extracellular space or the perivitelline space, and it make a mechanical barrier to entry of the sperm. Second thing, the actual substance that is released by these cortical granules have a certain enzyme. If we become more precise, it has a protease and glycosidase enzyme. That enzyme will ultimately denature or alter the chemistry of ZP3 protein that is present over the zona pellucida. So there are no more ZP3 available for the other sperm that they want to enter inside the oocyte. So no more ZP3 receptor, so no more sperm get attached to the zona pellucida, and less chances, or it nullify the chances of the second sperm entry. So let's get written down whatever we are discussing up to that level. So when the sperm membrane fuse with the olemma, it depolarizes the olemma or the oocyte cell membrane, and it stimulates the release of the intracellular calcium in the ovum, and it promotes the exocytosis of the cortical granules that is situated superficially to the olemma, right? And that cortical granules will inhibit the polyspermy by mechanical barrier, and by that it block the second sperm entry. Now the second thing, so block that occur at the two level. First level we already discussed, the cortical granule will fuse with the olemma, and olemma or the oocyte membrane is actually replaced by the cortical granules, so sperm has no affinity to bind with olemma, right? Now second thing is that the granule itself release the protease enzyme, that is protease and glycosidase. Glycosidase will alter the ZP3 and protease will eventually degrade the ZP3. That thing is known as zona block polyspermy. The prevention of a sperm to attach with the zona pellucida by release of the protease and glycosidase, it is known as zona block of polyspermy, right? So blocking of the polyspermy will work at two levels. First is the cortical granular level, second one at the zona pellucida level. Now what are the changes take place in the gametes? Now once the sperm entered inside the ovum, what are the changes which take place inside the ovum? What are the changes that is take place inside the sperm? So first we are discussing about the changes in the ovum. Obviously, as I told you in our ovulation time, there in our ovulation lecture, is that after the second meiotic division will be completed once there is a fertilization. So obviously second meiotic division is completed. So once the sperm has entered inside the ovum, oocyte divide to form a mature ovum and second polar body is expelled, and it complete the second meiotic division, and the nucleus, that female nucleus, it is known as now female pronucleus, and it has a 23 chromosomes. Now what are the changes that is taking place in the sperm? Once the sperm entered inside the ovum, actually the sperm head is eventually entered inside the nucleus, the ovum. So on entering the ovum, sperm has to change. Its head swells and form a male pronucleus, and this male pronucleus will contain 23 chromosomes. Got it? Now how the zygote form? It is rather very simple thing. Female pronucleus and male pronucleus, when they are fused together, they make a zygote, right? So male pronucleus, it has a 23 unpaired chromosome. Female pronucleus, it also has a 23 unpaired chromosome. So when they fuse, that actually form 46 paired chromosomal structure and fertilized ovum. That fertilized ovum, that having 46 chromosome, it is known as zygote. It is simple. And this fertilization is actually taking place over the ampulla of a fallopian tube, or the dilated portion of a fallopian tube. Got it? Now the fertilization is taking place. Now how the fertilized ovum will reach to the uterus? Let's discuss that thing. Transportation of a fertilized ovum. So this is the beautiful diagram. So at the time of the ovulation, ovum will release from the ovary and it is expelled inside the peritoneal cavity, and this ovulation, after the ovulation, the ovum along with coronary radiata will eventually capture by the fimbrial end of the tubule, this uterine tube or the fallopian tube. Now once the ovum get trapped or get catch by this fimbriated end of a fallopian tube, there is a peristalsis occur. This reverse peristalsis will eventually make sure that the movement of a ovum will be towards the uterus, and once this oocyte will reach to this region, that is the ampulla of a fallopian tube, and if the copulation occur, if the sexual intercourse is taking place, and somehow the sperm will eventually enter inside the uterus, and it will traverse through this portion of a fallopian tube to the isthmus portion, then interstitial portion, then it eventually entered and reached to the ampulla of a fallopian tube, and the fertilization is actually take place over the ampulla portion of a fallopian tube. So you see, the ovulation will take place about 14th day of the the ovarian cycle, 14th day of the ovarian cycle. So in 1 to 2 days fertilization will take place. So fertilization is actually occur about 15 to 16 day of menstrual cycle, or rather 1 or 2 days after the ovulation. So the lifespan of the actual ovum is 1 to 2 days, and after that, once the fertilization will take place, the implantation will occur after 1 week. So what is implantation? Implantation is nothing but attachment of a fertilized ovum to the uterine endometrium. So that implantation will take place after 1 week or the 6 days, right? So fertilization will take place at the 15th or 16th day of the ovarian cycle, and after 1 week it will reach to the endometrium, it will attach to the endometrium. Alright. Now as the zygote form, it start dividing, right? So the first cleavage, this is the second cleavage, and there are 16 cell stage will reach by the third day of the transportation, right? That 16 day cell, it is known as the morula. Got it? This one is the morula. 16 cell stage, it is known as morula. Once the 16 cell will appear, 16 cell stages appear, then after that, after 42 hours or after 3 days, during the day number 4 and day number 5, this morula will develop a fluid cavity inside it. Once the fluid cavity will appear, now the morula is known as blastocyst or blastocele. Got it? Now, so let me repeat one more time. Ovulation will take place at the 14th day of a cycle. After that, 1 or 2 days, the fertilization will take place. So, fertilization will occur at the 15 or 16th day of the menstrual cycle. After 6 days, the fertilized ovum will reach to the endometrium and implantation will occur. Now, during this transportation, at the 3rd day, the embryo will reach to the 16 cell stage, that is the morula. After that, 4 to 5 days, there is a fluid filled cavity will appear, and now the morula is known as blastocele or blastocyst. Got it? Now write it down, that thing. Fertilization is actually occur on 15 or 16 day of the menstrual cycle. Implantation will occur at the 6th day later, and first 2 division will take place about 2 days. After that, embryo reach to the 16 cell by 3rd day, and that 16 cell stage, it is known as morula. Now during day number 4 and day number 5 transport, there is a appearance of a fluid inside the morula and the cavity will appear. That cavity is known as the blastocele, and embryo is now called blastocyte. Now the implantation. Now as I told you that the 16 now the implantation. Now as I told you that there is a 16 cell stage, it is known as the morula, and once the fluid filled cavity will appear inside it, now it is known as blastocyst, that is present at the day number 4 and day number 5. Now once the fluid filled cavity will appear, you can see that the cell that is actually divided into the 2, right? This one is the inner cell mass that is situated over here, and another one is the outer cell mass, that is outer layer, is known as trophoblast. And the trophoblast, so basically this entire lining of cell, it is known as trophoblast, and this cell that is situated inside this trophoblast, it is known as inner cell mass. And the trophoblast that is situated near to this inner cell mass, it eventually make an embryonic pole. So trophoblast layer near to the embryonic pole, that is that layer of the trophoblast that is situated near to this inner cell mass, this portion is basically made an embryonic pole. So this embryonic pole is eventually attached to the uterine endometrium at the time of the implantation. Let me show you one more beautiful diagram so you can easily understand. This is the uterine wall, this one is the blastocyst, this outer layer, it is known as trophoblast, this lighter blue portion is the inner cell mass, and this portion that is present and eventually come in contact with the uterine wall, this portion is known as embryonic pole, and this trophoblast, it is known as embryonic pole. Got it? So eventually this embryonic pole region of the trophoblast and the cytotrophoblast, the inner cell mass will eventually made the placenta, right? This trophoblast that is situated outside, it is known as in situ trophoblast, that we discuss in our next slide. So this trophoblast that is present over that, it will differentiate into the 2 layer. One is known as the cytotrophoblast, another one is known as the in situ trophoblast. Let's discuss about the placenta. This is the actual formation of a placenta. So at the time of the implantation, trophoblast will differentiate into the 2 layer. One layer is known as the inner cell layer, it is known as cytotrophoblast. Outer layer, it is known as the in situ trophoblast. Got it? And the in situ trophoblast that is situated or make the outer layer, it has a 3 function. It attached to the endometrium or the decidual. Now endometrium of uterus after pregnancy, it is known as decidua. Decidua is nothing but endometrium, after fertilization and implantation. Now, and this decidua has a lipid rich cell, right? So that decidua will provide the cholesterol to the placenta in order to synthesize steroid hormone. So syncytial trophoblast perform three functions. One function is the adhesiveness. It attached to the endometrium or the decidua. It invade the endometrium. It actually buried inside the endometrium, and finally it produces the endocrine function. It is produce the steroid hormone. So you might be thinking, what is the function of cytotrophoblast? Cytotrophoblast make a machinery that will ultimately make this structure, that is villus, chorionic villus, primary, secondary chorionic villus, and it will provide the blood to the fetus. Got it? Now let's discuss about the function of the placenta. Now, as far as the function of the placenta is concerned, it is a various function like metabolic function, then exchange function, then endocrine function. It will release certain hormones, and protection of a fetus. Now we are going to discuss one by one. Now, as far as the metabolic function is concerned, the decidual cell, it is a highly lipid laden cell, and it also has a high in glycogen, right? And placenta make new cholesterol and other lipid. Alright, this is the metabolic function of a placenta. Now, as far as the exchange function is concerned, that exchange function is the simple diffusion, then the facilitated diffusion, then the active transport, pinocytic process, and the other transport. We are going to learn one by one. Let's discuss the exchange transport. And one more thing I would like to forget in the previous slide, the maternal cholesterol and lipid are not able to cross the placenta. So placenta, they have to make their own cholesterol and lipid in order to make a steroid hormone. Got it? Now let's discuss the exchange function. All the type of the exchange, it is utilized by the placenta. First simple one is the simple diffusion. Now, as far as the simple diffusion is concerned, gas is basically the carbon dioxide and oxygen will be transported via simple diffusion. Facilitated diffusion, basically the glucose via the glucose transporter will be diffused by the facilitated route of the diffusion. Then the active transport, basically the raw material of the muscle, amino acid, will eventually enter inside the fetus by secondary active transport. It will cross the placenta by secondary active transport mechanism. Now, pinocytic process by immunoglobulin G from the maternal circulation will enter inside the fetal circulation via pinocytosis. Now, the other transport. Now, what are the other transport? Now, this is the diagram of the pinocytosis. Now, what are the other transport mechanism? Let's discuss the other transport mechanism. First thing first, the maternal and fetal RBC exchange. Now, maternal fetal RBC get exchanged by making small breaks in the placental membrane. Certain virus will enter inside the fetal circulation. That virus is the cytomegalovirus and the rubella. Certain bacteria will eventually enter inside the fetal circulation, that is syphilis. The name of the syphilis is the Treponema pallidum. Let me show you a picture of the Treponema pallidum. It is the bacteria of syphilis. It is a spring-like structure. Then the parasite, basically Toxoplasma gondii. That is the parasite will eventually enter inside the circulation, fetal circulation. And the certain toxic drug that will enter from the maternal circulation into the fetal circulation, it is known as teratogenic drug. Let me give you an example, that is tetracycline. Alright. So, as far as the bacterial infection is concerned, only syphilis and Listeria monocytogenesis. Remember these two bacteria. One is the syphilis, another one is the Listeria. Both are capable to cross the placental barrier. Now, the most important one is the oxygen transport mechanism. Now, as far as the oxygen gradient is concerned, oxygen diffuse along the pressure gradient. So maternal circulation that reach to the placenta has a oxygen partial pressure of oxygen is 50 mmHg, and fetal side it is 30 mmHg. So it is very low gradient, right? And yet we have to provide a sufficient amount of oxygen to the fetal circulation. So this low gradient is a very serious problem. This serious problem, how can we deal it? We have to deal it with two facilitatory effect or two beneficial effect. One is the double Bohr effect, another one is the fetal hemoglobin. Now let's discuss the fetal hemoglobin first. Now this fetal hemoglobin, this is the diagram of the oxygen dissociation curve. It compare the adult hemoglobin with the fetal hemoglobin. You guys are familiar with this diagram. Fetal hemoglobin has a higher affinity to bind with the oxygen, to bind with the oxygen, and as compared to adult hemoglobin, right? Why the fetal hemoglobin has a higher affinity to bind with the oxygen? The answer lies is that it has a 2 gamma chain instead of the 2 beta chain. The 2 gamma chain has a less affinity to bind with 2,3-DPG, so there are less chances of the right shift of that curve. So if the 2,3-DPG is basically designed for right shift of the curve or to decrease the affinity of the hemoglobin to bind with the oxygen. Got it? So one thing is that fetal hemoglobin has a capacity to bind more amount of the oxygen in less partial pressure. Got it? And the next one is the double Bohr effect. Now what is double Bohr effect? Now in order to understand the double Bohr effect, you have to understand what is Bohr effect, right? So let me revise you a little bit from the respiratory system. This is the graph showing oxygen dissociation curve. In the x-axis there is a partial pressure of the oxygen is written, PO2, and over the y-axis there is a hemoglobin saturation will be written. 100%, that 100%, it suggests that 100% oxygen, 100% hemoglobin molecule will bind with the oxygen, right? So this line, the situated between the two dotted line, this formed line, it is the normal HB dissociation curve. Now once the affinity of hemoglobin to bind with the oxygen decrease, let me repeat one more time, if the affinity of a hemoglobin to bind with oxygen is decreased, this entire curve shift to the right. Now in which condition this shift to the right? Basically increase temperature, increase partial pressure of carbon dioxide, and decrease pH. Now the important thing that is in this discussion is the partial pressure of carbon dioxide. So whenever there is a higher amount of carbon dioxide, this carbon dioxide will eventually leads to release of the oxygen from the hemoglobin, and it will leads to shift to the right curve, shift to curve to the right side. Now on the other word, if the affinity of a hemoglobin to bind with oxygen is increased, then the entire curve is shift to the left side. Where this situation will occur? Whenever there is a decreased partial pressure of the carbon dioxide. Got it? So what is the Bohr effect? Bohr effect is that this right shift is the Bohr effect. Whenever there is a higher amount of the partial pressure of the carbon dioxide, what it will do? It will decrease the affinity of hemoglobin to bind with the oxygen. So oxygen is getting released. This is the Bohr effect. Let me write it down for you. What is the Bohr effect? Whenever the partial pressure of the carbon dioxide rise, it decreases the affinity of the hemoglobin to bind with the oxygen, and oxygen will eventually release. Now in our circulation, where this Bohr effect is seen, this Bohr effect is actually seen at the periphery, at the level of the cellular level, right? So cell will eventually metabolize and it make more amount of carbon dioxide. So the partial pressure of the carbon dioxide is higher at the periphery. So as the blood will enter along with the hemoglobin, that hemoglobin will expose to the higher amount of the carbon dioxide, and whatever the oxygen that is attached to that hemoglobin is eventually released. It is known as Bohr effect. Now if you see the other way, if the partial pressure of the carbon dioxide fall, then it will increase the affinity of the hemoglobin, then to bind the oxygen, and basically oxygen will bind with the hemoglobin, right? So reverse is also true. What is reverse to the Bohr effect? Whenever the partial pressure of the carbon dioxide fall, it will eventually increase the affinity of the hemoglobin to bind with the oxygen. So eventually oxygen will bind with the hemoglobin, right? It now just pay attention what happens inside at the level of placenta. See what happened inside at the level of placenta. Remember my word. So basically the fetus will made the metabolic waste product, right? It also made more amount of carbon dioxide. So at the placenta, at the level of the placenta, fetal circulation will carry the higher amount of the carbon dioxide or more amount of the carbon dioxide. So this carbon dioxide will eventually diffuse from fetal circulation to the maternal circulation. Got it? Carbon dioxide will diffuse from fetal circulation to the maternal circulation. So whatever the partial pressure of the carbon dioxide that is situated inside the fetal blood is decreased. So fetal blood become more alkaline, or in other words, pH of the fetal blood will increase, right? So what happens to the HbO2 curve? HbO2 curve will eventually shift to the left. So this hemoglobin oxygen hemoglobin dissociation curve will shift to the left in the fetal circulation at the level of placenta. Now if you see this picture from the maternal side, so more amount of the carbon dioxide will eventually enter inside the maternal circulation. So the blood that is present inside the maternal circulation become more acidic. So if the blood become more acidic, more amount of carbon dioxide that is present inside the fetal circulation, from the fetal circulation to the maternal circulation, there are more amount the maternal blood will eventually expose to the more amount of the carbon dioxide, right? So once the maternal circulation will expose to the more amount of the carbon dioxide, the HbO2 curve of the maternal circulation will shift to the right side. Got it? So at the level of the placenta, let me repeat one more time. At the level of the placenta, fetal circulation will eventually lost the carbon dioxide, or carbon dioxide will transported from fetal to the maternal circulation, or the carbon dioxide getting diffused from fetal to the maternal circulation. It will eventually leads to fetal blood to lost the carbon dioxide. So HbO2 curve in the fetal blood will shift to the left, and as the maternal blood will gain more carbon dioxide, so maternal HbO2 curve will shift to the right. So as if we talking about the Bohr effect at the level of the placenta, it will operate in the two direction, right? One direction in the maternal circulation, it will right side shift. Another direction in the fetal circulation, it is left side shift. It is known as the double Bohr effect, and this double Bohr effect will eventually provide enough amount of oxygen to the fetal circulation, obviously along with the fetal Hb. So let's write it down in a very simple term. If you understand that thing, let's write it down. Carbon dioxide and waste product from fetal blood will diffuse into the maternal blood, right? So loss of a carbon dioxide makes fetal blood more alkaline. So HbO2 curve will shift to the left. This HbO2 curve is about fetal circulation or fetal blood. Now as the carbon dioxide is increase of the maternal blood, it makes the maternal blood more acidic. So HbO2 curve of a maternal blood will shift to the right. So Bohr effect will operate in one direction in the maternal blood and the other direction in the fetal blood. It is called double Bohr effect. Did you understand? That's it for our today's lecture. In next lecture we are going to understand the endocrine function of the placenta.